Hello, everybody. My name is Gail Morris, and I'm the coordinator of the Southwest Monarch Study. I want to welcome you here tonight, and thank you for sharing your time with us. We'll be sharing some information about studies we participated in with the new radio tags, and we'll tell you more about that as we get into our PowerPoint. And Dr. Rutowski is here with me. You'll be hearing from him throughout the PowerPoint as well. And we'll have you meet our board members after the meeting as well. So let me bring our PowerPoint up and begin. So we are part of the Southwest Monarch Study, which was started back in 2003. Chris Klein actually started the study at Boyce Thompson Arboretum, and I know he's signed up for this as well, and I'm hoping we'll be hearing from him later today if he is. We have been tagging since 2003, which was advantageous when the issues of concerning monarch populations started coming into play, because we already had data from our earlier tagging and continue to do so today. So we'll jump into our program here. I'm going to do a few introductory slides only for people who aren't familiar with it. I'm assuming everyone's familiar with the basic monarch life cycle, that in general, an egg to a new butterfly takes about a month. It is very temperature oriented. Colder temperatures will make that take longer. It can speed up a little when it's warmer, and if it's too hot, it's not successful at all. Breeding monarchs, remember, live about 2 to 6 weeks, and fall migratory monarchs are not breeding and can live up to about 7 months. The speed of this process is determined by temperatures, and which can be really challenging in a dry climate, because our extremes of temperatures from day to night are much larger than what we see in humid environments. So tag monarch recoveries that we've had, you can see our map has shown that monarchs go to both Mexico and to California from many parts of the Southwest. The red lines were actually reported in our first paper. Since then, had we stopped, we never would have known all the other flights in the Southwest. And you can see some of the monarch flights from Utah, and some of the people who are joining us tonight, some of them have come all the way down into the Phoenix area and been seen here. Others have gone to California. So it's undetermined yet what's happening with Utah, but they may also go in most directions. And we're excited to have them tag as diligently as they had the last few years to help get more information about that. In addition, we have learned through our study that some monarchs spend the winter in the lower deserts. And we know this through tagging. We're used to tagging helping us with a migration destination, but it can also help us see that they're staying in one location by their longevity. And sometimes we can find these monarchs still here 3 to 4 months later, depending on when they were first tagged. One thing we have used to help us determine the time to start trying to do the radio tagging was going back to the angle of the sun on the day these monarchs were tagged that were recovered in California or Mexico, and tracking them by their sun angle. And that gave us a window of time that we know the peak migration comes through each area. And very interestingly, monarchs that come through late are never seen, are never recovered. We've only had, I believe, 3 that were tagged in November that were ever recovered. Otherwise, they stay in the local areas. If we take these tag, and you could see on this graph here, the yellow have gone to Mexico. These monarchs that are marked in blue have been recovered in California. Originally, we thought there was a time break, like at one point, monarchs went to California first and then to Mexico. Now we know it's independent. And again, had we not stopped tagging, we wouldn't be aware of that today. If we take those sun angles in 3-degree buckets, so to speak, you can see we get a bell-shaped curve for each area. So we can tell where the migration starts in low numbers and then peaks and then comes back down again. And these November ones are the ones that we're talking about that are right in this end of the migration period. And that helped us come up with the peak migration window that works in years where there is not excessive heat, which has been a challenge for us the last year or two. But the interesting thing is if they come in late, again, we don't get the recoveries that year. So these were the windows that we started using when we started looking at using radio tags. And the one that really got us thinking about this again. We have been talking about using radio tags for years and years and years. When I was on other committees, I was always intrigued by the studies that were going on. But the biggest challenge always has been the weight of a radio tag. And then this one happened. Denise Gibbs, who lives up in Cornville, outside of Camp Verde and Cottonwood, tagged a monarch, and this was one of the November ones in early November, and it was seen in San Diego, California. But look when it was seen, it was March 20th, so it was a spring sighting, which are unusual of tagged monarchs. And it got us thinking, like, okay, did it spend the winter in San Diego? Or did it go down into Mexico and it's on its way back, or Baja? And we really don't know. It was found in a swimming pool, but alive. And the person pulled it out of the pool and contacted us. So we saw the Southwest flight of 343 miles, but we don't know where it really spent the winter. So that challenge came up, realizing that a monarch only weighs about half a gram in the East. But Western monarchs are thought to be smaller and lighter, but there really is not much data available about Western monarch weights. And can you attach a radio tag to a monarch to track its flight? But what about the weight of the batteries, as we mentioned, since they were always too heavy to be used? And how else could monarchs be tracked? And then Cellular Tracking Technology developed a new BluMorpho radio tag that weighs only 0.06 of a gram, and it uses a solar panel instead of a battery that fits on the back abdomen of a monarch butterfly. So Cape May Science Center tested the BluMorpho tags in fall, and they were able to track the radio tags monarchs on a free app, your phone app, since it works on Bluetooth on their smartphones. And they were detectable as long as they stayed in the region, but they lost their signal as the monarchs migrated out of the area. Then Cellular Tracking Technology developed a longer range tracking app for Modus towers that could be retrofitted with a sensor, and it's a wider area that it could detect these tags, which were about 2 kilometers or more. And then they developed a smaller home backyard Terra unit and a handheld scanners that could also be used for detection for a distance of about 1 kilometer. It's about a little over a half a mile. So that made it easier to start seeing if we can have long range tracking instead of just local tracking. This is the Project Monarch app. Many of you may be already familiar with it, especially if you're on the East Coast. The app is free to download for all phones. It's very simple to use. You just basically follow the directions of hitting you sign up for the app like we do with most. You launch it, it comes up with this screen, and you just hit start scan. And if any radio tags are in your area, they'll start coming up, and you can see some of ours that started popping up as an example of what they look like when they come up. And then you just hit upload, and it uploads it so it's all recorded. So Xerxes did a pilot test of 3 monarch radio tags in Vandenberg, California, by one of their working groups, and it was successful, and they were able to track them within the grove for about 4 days. It was a year ago winter before the population dropped as much as it did this last year, and all radio tagging was put on hold. But we kept having more questions. This was exciting to hear, and it was wonderful to see that they were able to attach them. This is how the radio tags look when they're sent to you on this thin metal line. You cut them off right here, and you attach them here. You can see the monarch on the back of this monarch here. So we had a lot of questions still, like how long would these tags stay attached to a monarch? Could they still fly high in thermals if they were flying? Could they mate in spring? There were just so many questions that we didn't have the answers to. So we wanted to determine the feasibility of using the BluMorpho radio tags in the desert conditions in the Southwest. Remember the high temperatures we get here and the extreme dryness we have. So we reviewed what Xerxes had done and developed guidelines for monarchs that would be suitable to be radio tagged during the fall migration this last fall in October of 2024. We purchased 20 radio tags through a very generous donation that was made to us. And we decided that 10 would be used in the field for wild monarchs. And then we applied to see if we could do overwintering research at the Desert Botanical Gardens during the winter months when they were not using their butterfly pavilion to possibly test some things that we would not be able to do in the field, like longevity. Once they would leave an area on a wild monarch, we might not know, did the tag stay on or not, and how long it would do so. So after looking at others that had tested this, we decided that the monarchs we selected would have excellent wing conditions, which would indicate they're freshly clothed and likely migrators in October, with the weight of 0.35 gram for optimal success or above. Because we didn't know the weight of monarchs out here, we had to pick a cutoff weight to begin our looking for. So our first choice was partnering with Scottsdale Community College, and this became the place of our first monarch radio tag from Arizona. I think you could see why it might be attractive to a monarch. It's a little riparian area in the middle of a desert campus, and there's a lot of mist flower in that area. This is all mist flower, and oftentimes it's filled with queens and monarchs. And so that was the location that we chose. And we tagged the first one. We invited the instructors, the teachers, other people that were involved, the people at the campus to join us on their breaks between classes if they so choose. And Ron and others helped net a monarch, and together we went through the process of determining their weight, measuring their wings, and applying one of the radio tags. And this was our first one that was actually launched that day. And you can see it was on the 10th. It'll give you all the information on the readout from using this app. And it stayed there for about a week. on campus. So we would go every day and go back. But if we couldn't get back there every day, it was great because other people were also looking. You could see with 954 detections on campus and all these blue dots were the reports that were coming in on the app. So we could see that this particular monarch still stayed in the area around the moist riparian habitat rather than extending outwards. Oops, sorry about that. So, as we mentioned, we were hoping to be able to do more of a study at the Desert Botanical Gardens during this summer. I'm sorry, the winter months. Most of our 10 wild monarch radio tags were done at Scottsdale Community College. And once we received the go-ahead from the Desert Botanical Gardens to use their butterfly pavilion, we decided to plan our research. We covered the cost of the monarchs that were used and the 10 radio tags. Because of the low monarch population here that year, we decided to use farm monarchs. And we'll tell you a little bit more about the conditions to use this place in the next slide or two. We decided to use and compare 4 groups of 10 farm-raised monarchs in each group to monitor the effects of wearing the BluMorpho radio tags and if the tag adhesive adhered over time. We also looked to see if wearing a radio tag interfered with their flight, their longevity, or if there were any differences between males and females in how they responded. So we ordered these and received 50 farm-raised monarchs in glassine envelopes. We removed each monarch and then recorded their weight, sex, wing, and wing length measurements. This is the inside of the pavilion. If you're not ever been here before, you could see it has screen sides and is large. It has a lot of nectar plants. Desert Botanical Gardens very graciously upkept the plants, kept them watered, kept them fed. They kept sugar water available. So it was constantly maintained. Back here is a nursery where they have their immatures, and that was the room that we needed to use to set up the radio tag monarchs. And they even put up a sign. In fact, they just had this up today when I stopped by about that active research was in progress and mentions our partnership together. So again, here's the weighing of the tags of the monarchs on the scale, Ron's measuring the wings, so we could have all the information. The goal was to have 4 research groups of similar weights. So that wasn't affecting the outcome. Okay. My screen is not moving forward here. Okay. So 4 groups were created with comparable monarch weights. 10 would be the radio tag monarchs. 10 monarchs would experience the process to have a radio tag applied, but they were not radio tagged. We wanted to see if just being controlled for an extended period of time, which was about 6.5 minutes, could affect them. 10 monarchs would just have our blue Southwest Monarch Study paper tag on it, and 10 monarchs would only have a Sharpie marked ID on it as our control. So this was the process. So we needed to find a way to lay the monarch flat so we could apply the radio tag. Now, this is where the glue would go to attach the solar panel and antenna here. But of course, you know, monarchs don't come with nice little blue lines on their back where we know where to exactly put that. So you're looking for these white lines on their back and coming down the white line on their back. Surprisingly, we're using eyelash glue. And this one in particularly was highly recommended, so we stuck to their recommendation. You would apply some of the adhesive, wait 30 seconds. You'd also have some of that adhesive on the solar panel back. After 30 seconds, then you would apply it to the monarch and you'd have a 3.5-minute wait. Gail Steffi, if you are on our Zoom with us, because I saw you signed up, we took one of your ideas to restrain the monarch during the time. We saw many different examples. Sometimes people put a pair of scissors on their lower wings or a ruler. We saw an idea that Gail had using the top of a hanger, and it worked really well using glassine envelopes to lay the monarch down. They do not move. They do not fight. They are very calm, just like they are almost when we're tagging them. They don't really move a lot. I think out of all of the ones we've radio tagged over several of the research runs we've had over the last couple of months, it was only one that tried to wiggle their way out, and we had to kind of hold them. In Cape May, they actually hold the monarch by their wings and wait that time period. To me, we were a little more comfortable letting them lay still, and as long as they didn't seem to be bothered by it, this was a very successful way for us to go. Since the Desert Botanical Garden Butterfly Pavilion was a USDA approved space, we used that nearby room, the nursery that I mentioned to you, at the facility to implement the planned procedure. Then the other researchers would need to take the monarchs into the main pavilion. Because of USDA procedures, they needed to live out their entire life in the pavilion. Okay, so we could not take them out at the end. We radio tag the monarchs. We expose the monarchs to just the preparation process alone, the blue tags, or mark their wings as a control in one group at a time. Then the other researchers took them in mesh bags out to the main pavilion. They released them. They monitored them for a short time to be sure they could fly well. Linda's here today and Laura's here. Both of those people helped us at time to let them go and release them. And I'm sure would be happy to answer any questions about what they saw. So 1 to 4 researchers monitored the monarchs at least 2 hours every other day. And here you can see some of the monarchs here at the DBG, nectaring on the flowers. We had a sheet where we kept track of the monarchs that were present or were flying. We identified them. We recorded each visit, as well as the detection of the active radio tags on the app. So we not only made sure they were detected by the app, but that we actually saw them. Deceased monarchs were then collected and tracked as well. And Ron, would you like to share what we found?
Yeah, sure. Hello, everybody, and thank you for attending. So one of the key purposes of this experiment was to try and determine whether or not the radio tag itself or the process of radio tagging them or even the process of putting a paper tag on them alone would have an effect on their survival, their longevity. And so what we were able to do with the observations that we made over the course of the study was to come up for each animal with a rough idea of a pretty good idea of how long it had lived. Now, generally, this was done on the basis of visual sightings more than radio tag detections. because one of the things we learned is that sometimes the radio tag monarchs can die in a place where the sun's hitting them and the tag is putting out a signal. But they would obviously, their life would be over. So we came up for the 40 animals in the experiment, then from the data that we collected, the observations we made, I came up with an idea of what the duration of their life was. And what is plotted here is the how the number of individuals changed over the course of the experiment. The number of individuals that were still flying around were still alive. And so there are 4 lines here, one for each of the 4 treatment groups. The blue line is the line, essentially the survival of the individuals that were radio tagged. The orange line is animals that did not carry a radio tag, but were exposed to the process of restraint and handling that was involved in the radio tagging. The light gray line there are animals that got only a paper tag. And then the yellow lines were animals that were not had no tag put on them at all. However, they did have a very small mark that was quickly put on them with a Sharpie. So the idea here is that if, for example, the radio tags are having a very negative effect on the survival of the monarchs, then we would expect that that blue line should decline much more rapidly over the course of the experiment. That is, the number of animals living at each time period along the course of the experiment should go down much more quickly than for the other treatment groups. Now, I will tell you that overall, in the course of this experiment, we ended up with sort of typical lifespan, what I'm calling a lifespan of about 12 days, 12 to 13 days. And as you can see, there weren't really huge differences in these lines. It wasn't the case that the blue one went down quickly, and then, for example, the yellow one stayed up high and then maybe fell down toward the end. And this was confirmed, the idea that there were not significant differences in these treatment groups and how they survived over time was supported by an analysis of variance that I did on the data, in which we looked to see if there were any significant differences among these 4 treatment groups on average survival. And there were none. And so this suggests that, well, there's a number of things we can conclude from this. There's a whole bunch of sort of more anecdotal observations that we can talk about. As I mentioned one already, that the radio tags could often keep pinging even though the monarchs were dead somewhere, and sometimes we couldn't find them because, as you saw, the pavilion's pretty densely vegetated. But the key thing is that all of these tagging processes, the paper tagging, the radio tagging process, carrying these tags, didn't really, in the course of our experiments, seem to have significant effects on monarch survival. And this is great. I mean, this is good. It suggests that these radio tags, even though they increase the body weight of the monarch by about 10 to 15%, depending on what the weight of the monarch is, they don't really seem to be having negative effects on their ability to survive. And these 12, another thing that we are kind of excited about too in this fall study is that the monarchs lived about 2 weeks, and that would presumably give them enough time, even with a radio tag on their back, to get to aggregations in California and possibly even in Mexico. So these are all things that we're sort of continuing to think about and see what other conclusions we can draw from this study. But this is one of the ways we represent the data that we collected over the course of this study in the fall.
Right. And if I remember right, Ron, there were no differences between males and females with
Right.
longevity either.
That's right.
Yeah. Yeah, so again, like Ron showed us with the graph really well, there was no significant difference in monarch longevity in any of the four groups or within the sexes in each group. Um, and this has been a major concern, I know, of people who are uncomfortable with radio tagging. And to me, it was very comforting to see this information, as Ron pointed out. All the four groups, now we want to say that this is a small sample size. All right, this is a small sample size. In fact, at the end of this, we really wanted to do another round to see if it would repeat itself, but no one was able to find any um, uh, farm monarchs in January. I remember that. So we went that for that time, but we did say, you know, we'd like to be able to run an experiment again. And the other things that came out of this, like Ron pointed out, there was other incidental information that came out. We had a major heat wave in December here with record-breaking temperatures. And yes, we started seeing radio tagged monarchs were observed mating. Now, we only saw the males, and even in this picture, you could see the male on the top, the female underneath. I do not know if that was a radio tag female. I certainly didn't want to interrupt them in any ways. But they can successfully mate. And that was a question that came up for if they were tagged on the overwintering sites, what could happen during the spring migration. They had no problem finding someone to mate with. Now, the radio tag adhesive did stay adhered to the abdomen for most of the monarchs for the duration of the research, but two did fall off. And that kind of reinforces what Ron was saying. You know, when a tag falls off, we had one that would go off at 2 in the afternoon, every afternoon. Someone could detect it, but no other time. So we'd start looking where the sun was hitting at that time, and we never were able to find that one again. And again, that little caveat at the bottom in the note that the sample size was small, but there was no immediate lethal effect that occurred to prevent further testing of BluMorpho tags. But an opportunity came a little too sooner than we thought. At the end, near the end of the experiment that we just mentioned to you, Natalie Melankoff, who was our point of contact at DBG the entire time we were there, came up to me and said, "Hey, could you teach me how to do the radio tagging?" So we spent some time with me showing her how and teaching her how to do that. Because she didn't have radio tags, we used one of our radio tags. And so we named it DBG1, so it was theirs. And this is the pavilion over here. And this monarch thought he liked it better where the employees were and flew in this area and stayed in the employee area. But you could look that we did this tagging on December 5th. And this monarch stayed active until the 29th. So that is 24 days. I want to point out that this was a wild monarch that she brought over to tag. This was not a farm one, and it was loose at the Desert Botanical Garden. Notice it did not migrate. Remember how I mentioned earlier, if you see a monarch during the winter months, they're not going to migrate anymore. And that's exactly what hers did. So it kind of reinforced what we saw with paper tagging, that monarchs do stay in the area rather than migrating during the winter months when the nights are cold. So round two, we were excited when we heard from Natalie, where she came back and she said, "Hey, do you want to go back in the pavilion? But this time would like you to be in when the people are there." So this was going to be a different experience for us because we would have throngs of people that come through. Their numbers during the spring are between 1,700 and 2,000 a day that come through. And they could watch us do our monitoring, and we can see what happens. So we had this opportunity for round two. the Desert Botanical Garden offered us their radio tags that they had purchased, and we would return them if they were recovered. And we again covered the cost of the farm monarch butterflies were added to the pavilion on top of all the other species that they already had. We decided to modify our experiment a little bit. We would up the number of radio tag monarchs to 17. We would still have 50 farm monarchs. We would still do the 17 radio tag process only monarchs, but then we would use paper tagged monarchs. We would delete the opportunity to have the ones that had just ink numbers on their wings, because we were able to show earlier that there was no difference. We did come up with a better numbering system. We found that it was very difficult to see the little numbers on the tags, especially if they were on the top screens. And we came up with a series of dots and lines in black and red. The reds were the radio tags, the blacks were the process only, and red and blacks were the paper tagged ones. So if we couldn't read their tag number, we could often on that lower wing read the dots on it and identify them better. But this one had a very different results for us. Instead of coming down and feeding on the flowers like you saw earlier, these wanted to stay on the screens. And it didn't seem to depend on the weather. Even on warm days, on cool days, we always found them up on the screens, and they didn't really want to come down and feed. And so the results we had were much different. And Ron will explain what we found.
Yeah, what we noticed immediately from the outset of this experiment was that the animals were not living as long. And if you show the next slide, next, please, Gail. These were the mean lifespan or days since release in the pavilion for males and females in the 3 treatment groups that we ran in round 2 this spring. And you can see that the average lifespan was only on the order of 3 or 4 days, which was about a third or a quarter of what we'd seen in the spring, or I'm sorry, in the previous fall's experiment. And this is a puzzle for us. We ended up with no difference in treatment groups, again, like we had in the fall. No difference between the sexes and their lifespans. And so there's a way in which at a very broad level, we got the same sort of result, except that the lifespans were very, very different in duration, much, much shorter. And we're still puzzling about why this is. One of the things we consulted with people like Chip Taylor and others about what might be going on here. One of the ideas, one of the hypotheses that we decided to pursue was that these animals, we really didn't know exactly what condition they were in. And we thought, well, maybe we can help them along by feeding them before we release them into the pavilion. And so that then led to round 3, if you want to talk about that, Gail.
Sure, just introduce this here. So after everything happened so quickly, DBG came back to us again and said, let's do a round 3. And this time we're going to cover the tags and we're going to cover the butterflies. Could you come in and do the monitoring and reframe this and see what happened? We talked to Dr. Chip Taylor of Monarch Watch and actually walked around the pavilion with him on FaceTime so he could see what we were seeing and trying to come to some resolution. And Ron said, we thought, well, let's try to give them some sugar water first to feed them. We asked if we could order 25 farm monarchs because of the high rate of die-off. The last one we thought would take a smaller amount. would have one group that were radio tag monarchs that were fed sugar water first. We would have paper tagged only monarchs that were fed sugar water first. And then the third group would be paper tagged monarchs not fed as a control. But this time, only 15 monarchs arrived and only 4 were females. So we had to readjust everything we were doing. And that took a little while, but we still stuck to the 3 groups. You can see them sipping sugar water from the Petri dishes with cotton balls. And then when we took them into the pavilion, we actually, instead of just opening and making sure they could fly, we handpicked them up and we put them on a flower. And you can see they're all feeding. All of them stayed there. They were happy to feed. And we saw that for the next 3 days. They were on every flower. They were even coming down and sipping this sugar water. So there was a lot of activity. They were coming down. You can see some of the markings on the wings we were talking about. The V and the dot meant that was a radio tag. This is a red and a black dot. So that was our one that had no feeding ahead of time. But it was very easy for us to identify each of them. So then after 3 days, though, they turned to mainly hanging on the screen again. And so this is still in process. This is where we were at as of just yesterday. Today I saw one hanging from the ceiling, but again, I was just barely there, just walking through, and we'll let you know in the future what we find out. So
I would
like,
add just one thing, Gail, which
sure,
is that just
absolutely.
to give you a sense of where we are in this round 3 or this third experiment that we're running is going on right now. We're right at about a little over 1 week into it, I think. Is that right?
Yeah, it was last Wednesday that we
Yeah,
started.
yeah. And it does look like they're dying off quickly again, but we'll know. That's just our impression at this point. We'll know better in a few days or a week's time uh, when we finish this up.
Yeah. So, Ron brought up that question, can a radio tag monarch reach the overwintering sites? And yes, we really likely think it can because of an unusual occurrence we had where we could actually say, how long does it take? Because a lot of times when we're tagging in the fall, they're not discovered in California till around Thanksgiving, sometimes a week earlier, sometimes in January, and sometimes even at the beginning of February where we get a call saying, guess what we found? But in 2016, a freshly closed monarch tagged in Wilcox, Arizona, was seen 19 days later at Joshua Tree National Park in California, which is just right outside of Palm Springs during a NABA count. And that's a distance of 369 miles in those 19 days. So remember the one that I showed Natalie how to tag, and it stayed at the Desert Botanical Gardens for 23 days. So that gives us confidence, that while every migration flight varies, the outlook for future success using radio tags for tracking remains very positive. And it's exciting to consider pursuing this more. So many of you probably already heard this if you've been following anything about the BluMorpho tags. In December, a person in Florida with a backyard tracking unit system detected a monarch that was named Lionel, but he knew it wasn't in his backyard by the type of the signal. And he looked for a stronger signal and eventually found it in nearby St. Augustine, Florida. It was originally tagged on November 2nd at Cape May, and this monarch flew 726 miles before it was seen. And this was just on a little backyard unit, a Terra unit that was able to pick that up. So that's the first long-range radio tag recovery, which was exciting to hear about in the East. So detection's really going to be the key to sightings. And I think we all learned that using just our phones alone isn't enough, because once they leave our area, it's difficult to track them. And so we have been working in many ways to try to get the word out to see how we could expand the detection. And Modus towers with the 434 frequency can be outfitted with a sensor station to detect these BluMorpho radio tags. And that extends the detection range to 2 kilometers or 1.2 miles. And on this map, you can see the blue dots have the 434 frequency, but the purple are dual power ones and have the 434 and another frequency with it. So all those purple ones are potentials as well as the blues wherever you live, which are really exciting. And so I reached out to Blake Barbaby, who works with Point Blue and helps install and retrofit some of these. And he gave us this list of the ones that are ongoing in Arizona. There's some coming in around the Phoenix area, Flagstaff, Verde Valley, Horseshoe Ranch, Robbins Butte, which is on the Gila River outside Phoenix. Planet Ranch was my request. They were going to put one at Appleton Whittle Research Ranch. They still will, but I felt we needed to have more on the Colorado River since our monarchs are passing over that. And so we've worked with the Bureau of Reclamation. We've met with them the last 2 years in January and done workshops about the conservation along the Colorado River. And they were very excited to add it there and at the Cibolo National Wildlife Refuge further south near Yuma. Patagonia will be decided too soon. And then Apple Whittle will be up first on their list after the planned ones on the top ones. And you can see Whitewater Draw, San Pedro River, Buenos Aires. We are going to hopefully get much more tracking, not only on monarchs that might be going to California, but those that will fly to Mexico or stop somewhere in between. And to me, that's incredibly exciting to think of the potential of that in the future. So radio tags are really super expensive. They are $175 for one. When we bought our first 10, they were $150 for one. You do get a little bit of a discount, but even with your discount, I just wanted everyone to be aware of the costs of these tags. They are pricey, but like most new technology, we're hoping that in time this will come down to make it more affordable for everybody. This is that backyard terrace station that you've heard me talk about. This is something if a homeowner, it becomes a little more affordable. They actually track birds in the area and help you identify birds, and it'll track how many birds come through your yard. But it's also set up to track the BluMorpho tags. So they'll tag catch the tagged monarchs. If any bats are using the BluMorphos, you'll be able to pick that up as well. So this is another new piece of technology that's available. I can't thank enough people. I know this is just a sample of the many, many people who reached out to partner with us to help us along this process. Scottsdale Community College, the Desert Botanical Gardens have been outstanding. The Cape May Science Center and Project Monarch have helped us along the way. The Xerces Society has offered us some tags as well as helped us through the process. We had several anonymous donors step up and offer us the funding that we never thought would be possible. And the 400 dedicated Southwest Monarch Study monarch taggers that have given their time in the field that have led us to this point. It's been an exciting journey along the way, and we're looking forward and hoping we could do more. And this is our contact information, and we will definitely just open this up to any of your questions.
Gail, I have some questions that were posted to the chat.
Okay.
One by Penny you've already answered, which was the question about how much does a radio tag cost. But Penny also wondered, did the screen hangers hang on one particular side of the enclosure?
That's an interesting one. And Ron, I'm going to be interested in hearing your part. They do where the sun is. They're always where the sun is. There's a northwest corner seems to be the most popular with monarchs and the other species too, but we've seen them around the pavilion depending on where the sun is. And today they were at the very top. Ron, what's your thoughts on that?
Yeah. Well, I would just add that Nancy also had a similar comment slash question. Had those who observed the monarchs at the DBG noticed before that the monarchs tend to hang around the screens at other times? I've seen that behavior in other butterfly exhibits. And yeah, I mean, butterflies are very what's called phototropic, or they're attracted to light. And when put in a cage that has an unequal light distribution for whatever reason, they will tend to concentrate in those areas where light is abundant. And I don't know, there's a lot of variation among species in how well they adjust to these caged enclosures. Some of them just like the zebra longwings and the Julias in there, they just don't seem to care. But
Yeah.
they're sort of sub-canopy tropical rainforest species, and they're not too bothered by the shade. But more open field butterflies, it can be a problem. So, and it's frustrating to see them do that because it's not something you typically see them do in nature. So it does
Yeah.
make you wonder about that. There was
Yeah.
another question here. Oh yeah, where did you buy the this is from S. Where did you buy the farm monarchs for the study? Commercially raised monarchs may not be as fit as wild monarchs or might be more adapted to controlled environments.
Right, right. And actually the Desert Botanical Gardens purchased them and we paid them back for those first two rounds. I'm not at liberty to say exactly who they purchased them from. But yes, I agree that that is a distinct possibility. We don't know the age of the monarchs we use. We could see the one that Natalie bought was a wild monarch and was tagged and lived for 20 some days. Do you see what I'm saying? And ours in the pavilion were not living that long. But we needed a way to check on longevity. We had no other way to do that. When we're out in the public, we can't see how long they're living. We also can't see, do the tags stay on? So remember I mentioned in the first round, 2 tags fell off, but we were careful in round 2 and round 3 and no tags fell off, during that, at least the ones we're finding. We haven't found all of 3s yet. We're still recovering some of them, but no tags have fallen off that we're aware of in those. And I think that's another thing to keep in mind. When we first started doing this, Ron, with his expertise background, said, some monarchs may not survive the initial times you're learning to do this. We had no deaths, immediate deaths at least, of any monarchs, whether it's the wild ones with the people we've worked with or in the pavilion at the time because of anything we did with them, putting the radio tag or putting them through the process.
Yeah. The monarchs that as Gail mentioned, when we get these monarchs, they come in a box, and the first thing we do is weigh and measure them all. And if there were real significant issues with respect to their quality, you would expect them to be sort of smaller, both in body size and body mass, uh, than uh, than field caught monarchs, and that doesn't really seem to be the case. These ones that we get are in pretty good. They seem to be to us in pretty good shape, and we do make sure that when we release them into the pavilion, that the phrase I use is that they're capable of controlled, sustained flight. And so it's not the case that we release them and they just fall to the ground and lay there. They seem to be pretty capable.
You
Um,
know, Linda is here, and Linda was one of the people releasing them. If Linda, if you'd like to say anything about that?
Um, yeah, thanks, Gail. It didn't seem like they had any problem at all whenever I would bring them from the nursery into the pavilion. And, they would fly. I made sure that they could fly before I would walk away. So it didn't seem like they had any problem from having the radio tag attached.
yeah. Oh, there was also Robert Coffin asked, were round 2 and 3 both with spectators in the pavilion?
Yes, both of them.
And the answer is yes. They keep the exhibit up and running until Mother's Day.
Yeah.
So.
Are there any other questions? Do you have any concerns? I'd love to hear your thoughts. And it's okay to put your video on. I think it's neat to see all of you here, and your names, but I'd love to see your faces too. So. Hi, Simon.
So,
Uh,
Sid,
oh,
if you can hear me, hello everyone. This is Robert and Simone Coffin from Medford, Oregon, over on the West Coast.
oh, wow.
And
Yay.
hello there, Ron. Likes to see you smiling there.
Yeah,
And it's been a little while since we last all got together.
yeah.
So just to chime in, a couple of things since Gail, you offered or asked. Wonderful to see this. It looks like the first thing that you were trying to do is find out, will this work? Is it going to cause issues? Is it going to be a problem where we can't use it as a way to learn more about our monarch population? And it's really great to see that it appears that, hey, this could be a viable tool. Because let's face it, folks, tagging monarchs by hand, even though we love it, it's rather archaic, relative to the other ways that we track other natural wonders of the world and so on. So I guess a question that I would have is I guess, and I know you've probably thought about it, this was done all in a pavilion. And of course we hope, but there's a big difference between the migrating monarchs that need to not just hang out on a screen and hope and enjoy the sun for a while, and the food is right there. And of course, the next step might be, and that's the question is, would the next step be to somehow try to, first of all, use these 2 kilometer tracking devices, that distance, but also track monarchs that need to really book it, like, 30 to 50 miles a day to migrate. So I don't know. I'm sure you've all thought about it, and I just wondered if that's like something in progress.
I think the more Modus towers that get retrofitted with these will help. And you actually living in an area that's going to be getting a lot of retrofitting. Washington, Oregon, and Idaho are going to be getting large amounts of Modus towers in. There's a grant that went out, if it still goes through and everything, but it won't be really eligible for another year or so before they get everything up. So again, everything is kind of like waiting. We use 10 wild tags. I want to be real clear at the beginning. That was our first choice. I've never worked with farm monarchs before. This is my first time. You know, but we were forced to kind of because of the population numbers. So you're right. These are all really good questions, but the only way we're going to be able to start answering them is in these little increments, like what happens in this controlled setting. And then when we get out, it has the potential, you know, to work. In other words, we weren't sure. I remember Ron and I had several conversations. He was saying, you know, we don't know if they're going to just last 10 days and that's it, or they're going to fall off after 5 days. We've shown that that doesn't happen.
Yeah, I think these radio tags, they have promise in at least 3 arenas. One is for doing as we've been doing with at Scottsdale Community College and as happened at DBG from Natalie's release, is to sort of monitor the local movements of butterflies during the winter or other times of the year. And there is a group over in near Vandenberg Air Force Base who are using the radio tags to try and monitor movements of monarchs between aggregations and within aggregations as well. So I think they show a lot of promise that way. As the number of these towers goes up, the possibility of actually tracking monarchs as they move along their route is good. And also as we radio tag more and more, even though it does get expensive, you know, we're just so hopeful that some of these radio tagged individuals will show up in the overwintering aggregations in California. And because the beauty there is that with the radio tag, you don't have to lay eyes on a little tiny blue tag. No, yeah, you might not be able to pinpoint where it is up there, but you know, if a monarch has its wings open, the sun's on it, you'll pick it up, even if you're, you know, 100, 200 feet away. And that could be a very powerful thing, and we could learn quite a bit from that. So. Let's see. Oh, there's a bunch of questions here, Gail, or did you want to say something?
Yeah, I just wanted to add on to what you said. I'm in a working group that's doing some of that planning in California and everything, and they are making sure that some of these Modus towers and some of these smaller nodes and things that can detect them are set up at the major used overwintering sites. So they're going to be in place in the next, maybe as soon as this next year, if not the year after. So it's coming, you know, and that's what's so hard for us now. Once they leave our area, we don't know where they're going, you know, and it'll be exciting to hear.
Gail, here's a question from Gail, and this is, let's see. Oh yeah, were there other species of butterflies in the pavilion for rounds 2 and 3? The answer to that is yes. They get the butterflies that they get are mostly from the southeastern United States from suppliers there. So they get all these things like peacocks and zebra longwings and Julias that we don't get here, but they are there. Can the tags be reused? That's from Kathy Hart. Gail, you want to talk about that?
Yeah, we will find that out. We have collected all the radio tags off of the deceased monarchs, and we are in the process of getting the rest of the eyelash glue off and testing that. I wanted to talk to them at the people who produce them to make sure I don't use something on them that will affect them. But we have not recovered all the radio tags from everyone. Remember all those plants you saw? No matter how much Ron was on his knees and I was using all kinds of things to dig, we couldn't find them all. And then you'd think you'd get them all and you'd walk away and you'd come back the next day and there's some from the previous round that appear, you know, so it's a mysterious event. When we're in there, what we're going to find, but but hopefully we can reuse them. Yes.
Yeah.
But see, once we put them on and they go out of the range, we're not going to be able to see. That's that whole thing. So.
Let's see. Oh yeah, Angela asks, are there any Modus towers near me here in my area, Estrella Mountains?
Yes, Angela, that I thought of you when I was looking through that list. There's that one that's not too far from you in Goodyear. Which is because it's right along the Gila River, which is where you live. So that's going to be really exciting.
Yeah. Yeah, we may have already answered this. Judy Ann Hurst asks, if the butterfly dies and the wings cover the tag, can you pick it up? And the answer to that is no, typically. And so we have to, but hopefully you can see the butterfly. We dig around in the vegetation and hopefully recover them as best we can.
Yeah. If you're a gardener and you're used to digging in the dirt, it works. And actually, we've been using, have you seen those things, those little things with the handle that that have like a claw at the end? Sometimes we'll use that in the shrubs because you can move the shrubbery around and see what's underneath it and then pick it up.
Yeah.
Thank you, Angela. Okay,
So
do we have
I-
any other questions, or if anyone has anything after you leave tonight, don't hesitate to email us. We'll get you the best answer we can. We have a couple of our board members with us here. I know Laura isn't feeling well, so she's not gonna put her video on, but Laura Miller's here. Adrian Hopkins is here. We have a couple others that aren't here today. All of these
Yeah.
people help make this all possible. So just very happy to have all of you here to hear about this. And I hope this inspires you or raises some awareness. I realized this morning when I was trying to count how many monarchs have I radio tagged by helping people or doing it ourselves or for these experiments. And I realized it was 55 that I've tagged. And now we have it down real easy. We have a whole little process of how we get it down. But I remember those first couple times were very like I don't want to hurt anyone, including myself, you know. So, but it does work, and it does seem very efficient. It'll be exciting to see more research as it continues to happen to learn more.
Robert, did you have something else you wanted to say?
Yes, thank you. If I can, it fits really well with what Gail just said. I just wanted to mention, I guess a lot of the folks maybe here are from in Arizona, but I could be way wrong, and they're from all over the country. But I just want to extend kudos because for two reasons. One, for the work that you're doing, but I think a lot of folks know that are really interested in our Western monarch population as well as the Eastern. You know, where you're at, Arizona, you're like this, I like to call it in my presentations, you're like in the Bermuda Triangle of where the monarchs migrate or don't even migrate. Some migrate to the east, some migrate to the west, some stay local as you're learning, and some, like I saw down there, that stay down in Southern California and maybe pop over. So the fact that you're doing this in an area that's got even more questions about how our incredible monarch populations move around and hopefully survive. It's just great. So I just really appreciate not only the research that you're showing tonight, but also that this is being done in an area where we're going to learn a lot more about all of our migrating monarch populations. So thanks.
Thank you, Robert.
Thanks, Robert. A couple other questions, Gail, that I think are good sort of summarizing things. First of all, will the recording of this presentation be available for viewing later?
Yes, it'll take me a while to put it on YouTube, but I'll send a link out to everyone who registered. Okay.
Okay. The other question is that one was from Becca Berry. This one's from Torque. How can we help after hearing this radio tagging, about this radio tagging?
I didn't hear your question, Ronald.
It's how can we help after learning about this radio tag?
Keep your app on your phone whenever you're out. You know, it's right now, remember, all of us are pretty much dependent on this app. So whenever you're out, if you see a monarch, if you see something on them, put an app on and see if it picks up from wherever they're coming from. You know, it could be really exciting. You never know.
I would say this in response to that question that, yeah, the app is one thing. The other is that the radio tagging is not going to be a substitute at this point for the paper tagging. And that always there's always opportunities for people to help with that. And so that if you want to learn ways how you can help, visit any of the various Monarch websites, including Southwest Monarch Study's website, where there's information about upcoming tagging programs and all of this sort of thing. So
And we'll be posting that in the next month or so for fall.
yeah.
Do you know, just to tease everyone a little bit here, Arizona used to have the #1 amount of people tagging, but Utah has more taggers than Arizona does right now. So we need to be tagging. There's just too much happening right now. And we need to kind of, I know a lot of times it's a little scary to do. We show you how to do it and we have a lot of fun in the process. And you never know which monarch will make it to an overwintering site or just something very unique, like the ones that found out they were spending the entire winter here and they were alive 4 months later still. That's exciting information to learn.
Gail, a question from Rose. Remind us, which app should we be used for tracking the radio tags? And again, I think that's posted. The link to get the app is is available on the Southwest Monarch Study website, but it's called Project Monarch. So I think if you Google Project Monarch, you will end up in the place that will get you to that, to be able to download that app.
After when we send you the link with this, we can include that with it. And those of you that one of those backyard terror units, maybe that you love all kinds of things nature related, and that's something that would suit for you too. I'll send you just all the links and you could decide what you'd like to do. But the app is free. There's no charge for it. So please consider that. And you never know who's going to end up flying up into Utah this spring. So. Well, thank you, everyone. Ron, do you have any other questions? Thank you, Nancy.
No,
Thank you.
we'll let you know what happens, how this third round ends up.
That's true. We could include that in the letter in a week or so, too.
Right.
Thank you, everyone. Have a great rest of your evening. For some of you, I know it's really late, and I am so grateful you made the time to be here. And thank you, Ron, for all you've done to help us, and Adrian and Laura and everyone, Linda, everyone that's helped. Thank you. Have a good evening, everyone.
We'll see you at the pavilion tomorrow, Gail.
There you go.
Bye-bye, everyone.
Bye-bye.
Thank you.